Hello everyone, welcome to AS Biology with Dr. Demi. I am Dr. Demilade Fayemiwo, and on this channel I will be sharing the chronological order of the AS Biology syllabus with you to help you revise for your exams or to simply reinforce your understanding. I have taught biology for more than 2 years now, so I am using notes from my classroom to explain some of the difficult concepts, but covering all the chapters that are in the AS Biology syllabus. So if you simply want to reinforce your understanding, you want to revise, or you want to watch a couple of videos to see things in a different perspective, then this channel is certainly for you. And it would also be great if you want to interact with other students who are revising for their exams. You can simply post a comment and I'm sure other students would be happy to relate with you. If you have any questions, you're also welcome to post them in comments and I will get back to you with explanations as soon as possible. I am using notes from my classroom like I said earlier on, so this is going to be a narration of all the slides that I have prepared so far in the classroom. I hope you enjoy yourself. I hope you have loads of fun. Let us get into it. So to start you off, I'm going to first of all tell you about the textbook that we use in my classroom. This is a textbook that is accredited by Cambridge and it is by Mary Jones, Richard Fosbury, Jennifer Gregory and Dennis Taylor. It's the Cambridge International AS and A-Level Biology coursebook. The reason I love this book in particular is that the wording is very simple and easy to understand. So I encourage students very often to make sure they look into the textbook and not only rely on the things that are being said in class, because when you read from the textbook, especially after you've attended a class or even before, it helps to reinforce what you understand for a particular topic. What I have also done here, and I'm just going to use a pen to just highlight things when necessary, what I've also done here is show how I split the syllabus in my classroom. So for example, in term 1, my students cover 4 chapters. That's because term 1 is typically long, so they have lots of time to cover these chapters and to also learn how to do their laboratory experiments. In term 2, they cover another 4. Well, in term 2, it's more like 3.5, but we're able to make it work. And you can see there's a continuation there in term 3 of chapter 8, and then they cover the last 3 chapters. But by the end of the first year, all students would have completed the 11 chapters of the AS Biology syllabus. So if you're lagging behind at school or you just feel like you don't understand what's going on because you're being rushed, then this channel is certainly for you because I cover every single thing that you need to know. All right, let us start with our very first topic. So when we start AS Biology, we start with the basics. And that basic is that we start with the definition of what a cell is. It is believed in biology that every single thing that we see on Earth, when dismantled to its smallest, tiniest unit, will be found to be made up of cells. And as such, we define cells rather as the smallest unit of life. And cells are not just round structures that you see and they look pretty. Around them, you will see that they have membranes. So cells are not just hanging around and floating into each other. They have membranes, and these membranes are important because they control the substances that enter or leave the cell. And you will see why this is important when we get into chapter 4, for example. You begin to see the importance of these membranes and the role that they play in ensuring that cells are able to transport certain materials or even to communicate with each other. When we start with biology, something that's very important is microscopy. So when we speak about cells and we say that if we strip every living thing down to its smallest unit, we would find a cell. What many people don't know if they are new to biology is that cells are very microscopic. You can't simply look in the environment and just see a cell. You would have to put it under a microscope to observe it. And as such, that means that when we start this chapter, semester, we often introduce students to microscopes. And there are two key types of microscopes that you are expected to know. There is the light microscope, which is the one that you will typically find in your secondary school laboratory or your senior school laboratory. And there is the electron microscope, which is more likely to be found at a university. The electron microscope is a lot more complicated to run compared to the light microscope, and you usually have to have training to be able to run it. It uses software, it uses an electron beam to illuminate organisms so that they can be seen. The good thing with the electron microscope is that you can see a lot more with it. The light microscope, on the other hand, is a small desktop microscope, and you can see some organelles with it when you put a cell under it, but you can't see as much as an electron microscope. But for your level, you don't need to see that much. You simply need to see the things that are important for you to know for AS biology. I'm going to explain to you how the light microscope works so that you have an idea of it in case you haven't done this at school yet. So, how does the light microscope work? Before we even look into that, I want you to take note of the different parts of the light microscope. We call these parts over here, these two eye lenses, we call them the eyepieces or the ocular lens. And this is where you would typically look through as an observer to see whatever it is you're trying to observe. We call these lenses here the objective lenses. These objective lenses are for zooming in on an image or zooming out. So when we say zooming in or zooming out, it means that they can magnify an image. And what you will find is that you have one that is 10 times, which means that's 10 times magnification of the image. You have one that's 40 times magnification, and you have another one that is 100 times magnification. Something important for students to also know is that the eyepieces are always 10 times magnification. So it means that when you observe a specimen and you use a 10 times objective lens over here, the magnification of that specimen is actually 10 times 10, which means it is 100, because you multiply the magnification of the eyepiece lens with that of the objective lens. So that's something to just put at the back of your mind, because sometimes when you do some calculations in your laboratory exam, you will find that you are often asked these seemingly tricky questions, and it's very easy to get the answer wrong. This is called the stage over here, and on the stage is where you would put your slide. We call it a slide. It's a piece of glass that's rectangular, just like I have drawn there in red pen, and your specimen is always somewhere in the center there, and you're able to view your specimen by looking down at the microscope. At the bottom of that, you have what we call the condenser, and the condenser helps to concentrate the light that's coming from the light bulb. Now, something interesting I observe in classes with students sometimes is that they put the slide on, they turn on the microscope, but the condenser is not open so that the light is able to flow through. And then they scream, Dr. Demi, I can't see anything. It would be impossible for you to see anything if you're not allowing light to flow through the condenser. So the light needs to get through so that it can illuminate whatever it is you have put on the stage, and then you can view it here through the eyepieces. Another trick that you have to know with the microscope is that you should keep both eyes open when you're looking into the microscope. Students tend to close one eye, and if you close one eye, your observations are likely to not be accurate. So try to keep your eyes open. I know it can be a bit difficult. This here is for focusing, this knob, and you will see if you're able to do this in the classroom, that the more you turn either the tiny knob or the big one, that your image changes in how resolute it is, or in the resolution of it. So it means your image either becomes really blurry or it becomes more focused. So it is important for you to just bear that in mind. I have put the slides here, or the points here, to help you know the functions of the different parts of the light microscope, having discussed with you how the light light microscope itself works. So the eyepiece will focus the image from the objective. So think about it this way. Let's think of it working from the bottom to the top. You switch on the microscope here by the side. This is often the power button, and a light bulb comes on here. That light bulb then travels upward, or the rays of light travel upward, and they illuminate this image here. That is picked up by the objective lens, the size of the image picked up is dependent on the objective lens you're using. If you're using a 40 times lens, the image would be bigger. If you're using a 10 times lens, the image may be smaller, but might be more focused or sharper in resolution. Now that is picked up by the objective lens, and it goes up all the way over here to the eyepieces so that you are able to then see the image. The condenser lens, like I said, focuses the light onto the specimen, and you have the iris of the condenser, which is basically used to control the amount of light that you're getting. So that's just that little pointer over there. It helps you control how much light you're getting onto your specimen. But if you do this in the lab, I'm sure it's much more fun than what I have just explained. All right, the two types of cells that you are likely to observe, or basically the only two types that you will observe for AS biology, are the animal cell and the plant cell. Usually, students who are new to biology struggle to understand what the difference between these two cells are, but I often start students off with a spot the difference activity. What this activity simply does is to remind students, first of all, of the basic differences they can see by simply looking at these two cells. Now, what can you see over here? This is the animal cell on the left. The animal cell here is a round shaped or a spherical shaped cell that contains all of these organelles. You can see there it also has a nucleus at the center. We will discuss the function of the nucleus later. This is the nucleus, rather. It has a nucleus within the nucleus, and it has its mitochondria. It has a smaller vacuole that you often would not see under the light microscope. If you look at the plant cell, which is labeled plant cell here, you'll see that there is quite a bit of a difference just at first glance. The plant cell also has a nucleus. It also has a nucleolus, but then it has this big, wide open organelle, and this is called the vacuole. Sometimes you're able to see the vacuole, not sometimes, most of the time you're able to see the vacuole when you observe the plant cell. So it has a large vacuole, and that is usually for the storage of waste material or for the storage of water or nutrients, depending on what is going on within the cell. We often say that the plant cell has a large central vacuole. So with that said, I advise you to go into your textbook and to use the power of observation. Simply look at the images of a plant cell and an animal cell and ask yourself, what are the differences between these two when I look at them at first glance? What is it that a plant cell has that an animal cell does not? And what does an animal cell have that a plant cell does not have? If you're able to do that, even before you memorize the organelles or before you learn about their functions, it would help you very well in when you're doing observations with a microscope, because with the microscope you will see that the plant cells are very distinctly different, or rather are just distinctly different from the animal cells. So with that said, then we can ask ourselves what plant and animal cells have in common. First of all, they have a cell membrane. Remember at the beginning of the chapter, or at the beginning of the video, I said to you that cell membranes exist to help cells regulate what goes in and what they allow to leave the cell. So a cell membrane plays a very important function, not just in doing that, but also in communicating with other cells, something that we will see when we get to chapter 4. They also have a nucleus in common. If you remember the images we just looked at, we saw that both cells had a nuclei, they both had nuclei, which is the plural of nucleus. They also have cytoplasm. The cytoplasm is where all of the different organelles of the cell are floating. So think of it like a stew, and in this stew there are all these different pieces of vegetables or beef or chicken, depending on what it is that you're into. But that is what the cytoplasm simply is. It's like a soupy mix of all the things that you need within the cell. They also have mitochondria. Mitochondria play a very important role in providing energy for the cell. The mitochondria plays this role because it is called the powerhouse of the cell. And as you go deeper in biology, and that is beyond the scope of the AS syllabus, so this is at A level. At the advanced level, you will learn the processes that the mitochondria goes through in order to make ATP, which is the energy currency of the cell. They also have Golgi apparatus. Golgi apparatus are very important in the transportation of proteins, in the folding of the proteins that are made by the cell. And again, all of these will become more and more interesting as you learn more about biology. So don't stress too much. If it feels like, oh my goodness, this is just a lot, you will gain understanding as we go along. So here, can you tell the difference? Usually when I give students this, it is to help them see that sometimes in your question papers, the cells are not easily drawn for you to be able to see. They are not presented in color where you can simply look and say, oh, look at that, it's a beautiful animal cell. Sometimes they come out in micrographs that look like this. And so I use this to familiarize students with how the animal cell would look or how the plant cell would look if it was in a micrograph. Always pay attention first of all to the shapes of the cell. When you see a spherical shape, it means you are dealing, or something that looks like a spherical shape, it means you are dealing with an animal cell. And when you see a cell shape that kind of looks like a rectangle but not quite one, then you know that you are dealing with a plant cell. In other words, the plant cell has edges sort of compared to the animal cell. The animal cells tend to be continuous spherical images or structures when you view them under the microscope. Now, something big that you have to know, and this is important for the next video, is what we call magnification versus resolution. Now, if you take your phone camera today, you can probably zoom 5 times and everything would still be clear, especially if you use a very good phone. With some phones, however, the moment you start to zoom, what do you notice? That whatever it is you're focusing on becomes blurry. It is not as clear or as crisp as you would like it to be. This is why we say magnification versus resolution as opposed to magnification and resolution. Magnification is the number of times by which an image is bigger than the actual object. So if, for example, you start to zoom in on a dog that is far away, that dog, as you zoom in, the resolution might be compromised, but you have made the image bigger by zooming in. It's definitely bigger than the dog that you're looking at. So that is what magnification is. Resolution, on the other hand, is the ability to distinguish between two objects which are very close together. So this image of this dog over here, this is a very clear image of the dog, but as you can see here, this is not a so clear, distinct image. What has happened here is that the resolution has been affected, so you can't tell the distinct features of this dog by looking at it anymore. You can't see its eyes or its nose, which means that you've sort of lost your resolution or your crispness of this image as opposed to the previous image on the left. So, can we measure cells? Since we talk about magnification and these things that are bigger or smaller or more resolute, then the question becomes, well, how do we know when something has been magnified, and how can we tell by how much it has been magnified? Also, how do we just measure cells generally speaking? The formula for magnification is that you divide the observed size by the actual size, or most times we like to write it as I over A, where I is image size and A is actual size of the the image or the object rather that you're looking at. Also, something that you must learn here is the conversions or the units with which we measure. So there are 3 units that you're likely to come in contact with as you go through the syllabus. You're likely to come in contact with millimeters, micrometers, and nanometers. Now, when you want to go from millimeters to micrometers, all you have to do, it's taking a while here to write this down because I'm using a mouse. If you go from millimeters to micrometers, all you have to do here is multiply by 1000. Because from millimeters to micrometers, you take in an additional 3 zeros. If you're going from micrometers to nanometers, that is times another 1000 over there. So what this tells you then is that if you want to go from meters to millimeters to micrometers and to nanometers, then this is nanometers. It's just going to write this is an N. Actually, just going to erase that so you're able to see it better. So that's an N. So meters to millimeters would be times 1000. Okay. Millimeters to micrometers would be times another 1000. So I'm just going to write it as 10 to the power of 3. Micrometers to nanometers is times another 10 to the power of 3. So if you're going from meters to micrometers, it means that you're going to do 10 to the power of 3 twice. So that's going to be times 10 to the power of 6. I hope that makes sense. If you're going from meters to nanometers, it would be 10 to the power of 9 because you're doing 10 to the power of 3 times. And it's important that I show you that because on the next slide, I have an activity for you to do that you can do just to practice your conversions of these units because they become important once you start to do your microscopy experiment. So this here is an activity I often give my students to wrestle with, just to help them practice how to convert from different units. So you can see here that I have put different numbers on the different units, and what I would like for you to do is to convert them into the other units. So for example, 0.5 meters, what would that be in millimeters, in micrometers, and in nanometers? And you can do that for all the different numbers that are here. So it's just something nice to help you practice and to get familiar with conversions, because when you start to do the microscopy experiments, these become very important. As a matter of fact, if you don't use the correct unit in a calculation, you are likely to get the answer wrong, and that means you lose marks for something that is such a simple thing. So I don't want you to overlook this activity. Try to give some thought to it and to work through it, and I will see you in the next video where we look at this activity again and I show you the answers. Until then, have a good time.